You said something earlier about, oh, you know, you can tell this person is Chinese and this person is English. That's much easier to do. You said that earlier about, you know,
skull shape.
No, but you're just talking about phenotype in general. And I want to pull you up and say that's much easier to do outside of Africa than it is within Africa.
Redundant word salad says what? Regardless of his mealy-mouthed nonsense, we can, as a matter of fact, determine who is who and to which continent each belongs. Skull shape is indicative of phenotype. It doesn't take a genius. Really, it doesn't. Even though Andy prefers to live out of place among the lily-white British he despises instead of among his own in sub-Sahara, that doesn't change his ancestry or ours, nor does it make him or us any less recognisable in terms of origins. Geography, culture and language may change, but biology doesn't. Mongoloid, Caucasoid, Negroid, nothing difficult about these distinctions at all. As far as phenotypical variation inside Africa, a Koisan, a Pygmy, a Dinka, a Bantu, and of course a Moroccan, what does he mean it's hard to distinguish phenotype inside Africa? Anyone who studies populations and geography knows Sub-Sahara exhibits regional differences and is vastly unlike North Africa. Let's continue.
Because Africans show diversity even within small tribal groups.
I can tell.
Even within small tribal groups, they show massive, you will know.
Africa contains many distinct populations and therefore is not of a single monolithic type, hence why Africa is not and has never been a blacks-only continent. Nilotics, East Africans, Koisan, Pygmies, Bantu, Cushitics, Berbers, Egyptians, and perhaps a few more I haven't mentioned. These groups differ dramatically in phenotype, skull form, body build, hair texture, genetic lineages, and language families. But is Andy's statement even true? The claim that there is more genetic variation within populations than between populations originates with the Marxist geneticist Richard Lewontin, who first coined this interpretation in 1972. Lewontin carried out statistical analysis showing that genetic diversity is much greater within populations than between populations. His results indicated that 85.4% of diversity was within populations, 8.3% within races between populations, and 6.3% between races. This led him to state the following. Quote, "It is clear that our perception of relatively large differences between human races and subgroups as compared to the variation within these groups is indeed a biased perception, and that, based on randomly chosen genetic differences, human races and populations are remarkably similar to each other, with the largest part by far of human variation being accounted for by the differences between individuals." He goes on to state, human racial classification is of no social value and is positively destructive of social and human relations. Since such racial classification is now seen to be of virtually no genetic or taxonomic significance either, no justification can be offered for its continuance, end quote. This statement is wrong on so many levels. First, in the real world, forced integration has been destructive of social and human relations. This has been proven over the course of the last 60 years in the United States and since the dawn of time. Secondly, so much for Black Egypt, right? I mean, why bankroll your so-called Black Egypt position on a statement and source that rejects the reality, the entirety, the biology of race? Lewontin's statement is repeated by race deniers that ironically, hypocritically, push the Black Egypt fallacy. On one hand, ideological hypocrites like Andy and the like will tell you race doesn't exist, but on the other hand, ancient Egypt was racially black, and you better not forget it. Afrocentric inconsistency has skyrocketed off the charts since its self-serving, miserable inception. The contradictions are difficult to ignore. Inconsistencies are piled mile high for Afrocentrism, so much that it reflects a broad ideological problem rather than a scientific one. But was Richard Lewontin correct in his analysis? Was he correct in his statement? Lewontin's analysis was found to be faulty, and his conclusion has come to be known as, yes, you've guessed it, Lewontin's fallacy. This was first pointed out by Cambridge University statistician, geneticist, evolutionary biologist and professor Anthony Edwards in 2003 in the study titled Human Genetic Diversity, Lewontin's Fallacy. Edwards noted Lewontin's analysis was locus by locus, or each locus being considered independently of the others. Edwards states, quote, genes at a single locus are hardly informative about the population to which their bearer belongs. In other words, populations or races do not differ by genes considered independently, but rather by the many connections that exist between and among the genes, since genes are aggregated networks, end quote. What I find extremely disingenuous, hypocritical, and oh so self-exposing Andy, promoter of the Black Egypt lie, would rely on Richard Lewontin, a well-known race denier, as his authority. In other words, Andy is citing a source, referencing a statement that destroys his own claim, an act that is not only contradictory, but intellectually dishonest and transparently desperate, and not one single clapping seal questions it. So where is all this supposed greater genetic variation within populations than between populations? If that statement were as meaningful as race deniers insist, then we would not be able to distinguish human groups so easily at the genetic level. Yet modern population genetics can accurately assign ancestry from DNA with extremely high confidence. Here, various examples, in addition to Edwards, that dismiss Lewontin outright. Notice all these genetic studies state that populations cluster together. Ethiopians are more genetically similar to other Ethiopians than they are to non-Ethiopians. Bantus are more closely related amongst each other than admixed Bantus. North Africans are more related to Eurasians than sub-Saharans. Takakori is a better fit for Tafaralt than sub-Saharans. Celtic groups are more related to each other than non-Celtic groups. Levantines are more related to their Natufian predecessors. It means between population differences are clear and measurable, regardless of the misleading slogan repeated by activists who never explain the science behind it. Algorithms like admixture, structure, or PCA-based methods classify individuals into continental clusters with over 99% accuracy using as few as 100 markers. Clustering completely dismisses Andy's tired debunked cliché. If within group variation erased population differences, then principal component analysis would not show distinct clusters. Instead, individuals would be scattered randomly across the plot with no structure at all. But that is not what happens. Every major population clusters with its own group, forming clear clines and separations with other groups. If groups were not genetically distinguishable, PCA would be impossible as a tool, yet it works precisely because the differences are real and measurable. It's amusing Andy would feel so compelled to utter such a notion, such a cliché, such rubbish that disagrees with his laughable Black Egypt. You either accept race as a biological reality or you don't. There's no middle ground. Lewontin used his now overtly debunked analysis to attack racial classifications for social reasons. Yet Andy wants it both ways. He cites race denial arguments when convenient, then turns around and insists ancient Egyptians were definitively black. That is textbook cognitive dissonance, arguing two mutually incompatible positions without so much as realizing it. So no one is more related to someone in another race 20,000 miles away than they are within their own racial group or regional group or family group. Therefore, the nonsense that races have more variation within them than between them is a lie.
Show massive amounts of diversity, and that's perfectly normal for Africans, and that's not reflected in our Eurasian counterparts.
Bingo. Eurasians do not possess sub-Saharan genetic diversity for the simple reason Eurasians are not sub-Saharans. The out of Africa myth is swept into the dustbin of non-existent history. Eurasian genetic legacy has endowed us with our own distinct biological diversity, shaped by migrations, founder effects, genetic drift and the like produced patterns of variation unique to Eurasian groups. Indeed, Eurasians have their own distinct population histories, cultures, civilizations, skeletal morphology, genetics, phenotypes, ancient and modern, all very much unlike modern sub-Saharan populations. A major contributor to the high genetic diversity observed in sub-Saharans is the presence of archaic ghost ancestry. This is not the same genetic diversity expressed in Eurasian populations. Genetic studies have demonstrated that sub-Saharans carry DNA from extinct hominin groups that interbred with modern humans roughly 35 to 40,000 years ago, and this component ranges from 2 to a staggering 19%. Because these admixture events happened entirely within sub-Sahara, and because it took place 40,000 years after the supposed out-of-Africa fantasy, this ancestry is not shared globally. It is therefore absent in populations outside sub-Sahara. This 2 to 19% explains the much idolized diversity that Afrocentrics have ignorantly latched onto without the realization it is due to extremely divergent lineages, which did not in any way contribute to the gene pools of Eurasians or other non-African populations. Since this ancestry is confined to sub-Saharans and not shared with non-Africans, it increases internal differentiation among sub-Saharans. That's it. Extinct ghost DNA. This is the basis of Andy's argument, even if he never admits it. What he wants to say is that the degree of genetic diversity observed within sub-Sahara depends heavily on the specific population or tribe, with some groups or individuals showing substantially higher levels of extinct ghost population archaic admixture than others. And let me remind you, when the low 2% Neanderthal admixture was first identified in Europeans, Afrocentric racists attempted to use the finding as a point of ridicule, insulting, sneering, denigrating, projecting an all-out hatred of white peoples. But the irony couldn't be more brutal. Subsequent genetic research has demonstrated that sub-Saharans possess even higher levels, up to 19% ancestry, from an extinct hominin. These same racists now find themselves face to face with findings that make their earlier posturing look painfully premature. East Asians have 2 to 4% Neanderthal, Papuans and Aboriginal Australians have a 2.4% Neanderthal component in addition to a 4 to 6% Denisovan component. But you didn't hear Afrocentric racists denigrate these people for it. Our analyses provide support for a contribution to the genetic ancestry of present-day West African populations from an archaic ghost population whose divergence from the ancestors of modern humans predates the split of Neanderthals and modern humans. Did you read that? Predates the split of Neanderthals and modern humans. This is over 600,000 years ago. What archaic hominin population was living in sub-Sahara at that time? Here's some food for thought. Could it be Homo erectus, Homo ergaster, Homo heidelbergensis, Homo rhodesiensis, Homo naledi, or perhaps something else? We determined the posterior mean for the split time to be 625,000 years before the present and the admixture time to be 43,000 years before present. This means that a deeply divergent archaic hominin lineage survived in sub-Sahara long enough to interbreed with the ancestors of present-day sub-Saharans. In other words, these archaic hominins and early human groups in sub-Sahara were living side by side, and so these early groups were not yet modern. This was a localized sub-Saharan phenomenon, which is precisely why it boosts internal diversity within certain sub-Saharan groups. Importantly, this archaic component is absent in Epipaleolithic North African Ibero-Maurusians, highlighting that this ghosted mixture is not a pan-African signature and was not part of the ancestry of North Africans or Eurasian populations. So much for the We Was Everyone brigade. Archaic forms persisted in sub-Sahara until fairly recently. Up to 19% sounds like they continue to persist today. It is plausible that the history of sub-Saharan populations includes genetic contributions from multiple divergent populations, as evidenced by the large effective population size associated with the introgressing archaic population. This is precisely where haplogroup E becomes relevant. A North African branch of E1b migrated southward into sub-Sahara near the end of the Holocene. As the Sahara dried and the desert expanded, these populations became isolated in sub-Sahara and subsequently mixed with the local archaic hominin groups present there. Sub-Sahara demonstrates a mosaic of recent skulls. Perhaps an even more striking manifestation of the degree of structure in African populations and its time depth is the more recent evidence of mosaic fossils. Far younger fossils, such as the skulls found at Ishango in the Congo, dated 25 to 20,000 years. Those in Iwo Eleru, Nigeria, dated at 11.7 to 16.3 thousand years. Or those in Lukenya Hill, Kenya, dated 23.6 to 22.9 thousand years ago, display robust, archaic features that are outside of modern human variation. Do you see this? Outside of modern human variation, but also distinct from many Holocene specimens in Africa. Did you see this? Distinct from many Holocene specimens in Africa. And importantly, it states showing greater resemblance to specimens from the middle to the late Pleistocene in Africa, 100 to 300,000 years ago. Does anyone understand what is being stated here? The ramifications. It means these ancient people did not look like current sub-Saharans. The skulls and bones exhibit traits that current sub-Saharans today do not possess. Importantly, they did not resemble Holocene sub-Saharan populations as recently as 10,000 years ago. It means current sub-Saharan populations did not resemble populations 10,000 years ago or earlier. They were outside of modern human variation. In other words, they were so diverse, they were outside of current sub-Saharan variation. These individuals were not anatomically modern sub-Saharans at all. They carried archaic, highly divergent traits that no living sub-Saharan or non-African population expresses today. And what about post-Holocene sub-Saharans, the populations that emerged after 10,000 years ago? What became of them? Could they have descended from this earlier amalgamation? Sub-Saharan African populations form a well-differentiated cluster from the remaining world populations, because Eurasians do not share in this ghost DNA that sub-Saharans possess. Sub-Saharan African populations form a well-differentiated cluster from the remaining world populations grouped in two close clusters, that of Asian populations on the one hand, and that of North African, Southern European, and Middle East populations on the other hand. The old mantra about most genetic variation being within populations rather than between them has been debunked time and again, but ideological hardliners still cling to it as if the new data doesn't exist. Additionally, sub-Saharans forming a well-differentiated cluster apart from other world populations is proof evidence that we never was. Here, another study about the Ishango skeletal remains. Comparisons with large samples of late Pleistocene and early Holocene modern human fossils from Africa and Eurasia show that the Ishango human remains exhibit distinctive characteristics and a higher phenotypic diversity in contrast to recent African populations. What does this mean? This ancient sub-Saharan population exhibited more phenotypic diversity than modern sub-Saharans. Continuing in many aspects, as is true for the inner ear conformation, these early late Stone Age human remains have more affinities with middle to early late Pleistocene fossils worldwide than with extant local African populations. What does this mean? Well, it means the Ishango fossils show unique, highly variable physical traits compared to today's sub-Saharan populations. In several features, such as the inner ear, they resemble much older fossils from around the world more than they do modern sub-Saharans, suggesting that early sub-Saharan populations were more diverse and retained traits shared with ancient humans globally compared to living sub-Saharans. Sounds like into Africa, not out of Africa. Let's continue. Let's turn to the early Holocene sub-Saharan Nataruk specimen and examine just how different it is from modern sub-Saharan populations. Here we describe 8 mandibles from Nataruk, an early Holocene site roughly 10,000 years ago or 8,000 BC in West Turkana, Kenya, offering the opportunity of exploring population diversity in Africa at the height of the African humid period. In this paper, we examine the phenotypic variation of the people of Nataruk in order to understand their phonetic affinities with worldwide populations, and more specifically, with African populations. The comparative sample, listed by population and by sites in case of fossils, was chosen to cover a wide range of chronological and spatial variation. Chronologically, the range incorporates examples from the late Pleistocene 100 to 12,000 years, early Holocene 12,000 to 6,000 years, late Holocene 6,000 to 2,000 years, and 19th and 20th centuries. Geographically, the specimens originate from sub-Saharan Africa, North Africa, continental Asia, South Asia, Europe, Oceania 38, and Greenland. The recent European mandibles originate from England and from Eastern Europe in order to capture more of the European morphological variation. In sum, the Nataruk mandibles, while having a very clear modern human morphological pattern, show a number of features that are common in other hominin species, such as the Neanderthals. Such a phenotypic pattern has been described before in other early Holocene African populations, for instance, the specimens from Assi El Habiod in northern Mali and Iwo Eleru in Nigeria. The Nataruk mandibles are part of a continuum of morphological variation that links Pleistocene and Holocene populations from eastern and northern Africa to the almost complete exclusion of the recent African sample. Considering the phenotypic affinity of the Nataruk sample within both a worldwide and an African context, the analyses highlight the strong affinities that exist between Nataruk and other early Holocene populations from East Africa. They also show strong relationships with Pleistocene specimens from North Africa, Afalo and Taforalt, and more surprisingly, the older Nazlet Carter II specimen. These results place Nataruk within an East African forester hunter-gatherer morphological continuum in the early Holocene, which shares strong affinities with late Pleistocene fossils from northern Africa. Finally, it is interesting to note that neither the late Pleistocene North African sample nor the early Holocene Eastern African specimens, including the Nataruk fossils, show strong affinities to the specimens representing the extant or living African populations in our sample, as it has been pointed out in the morphological description of other late Pleistocene, early Holocene African fossils. These specimens tend to exhibit distinctive characteristics and higher phenotypic diversity than do extant living African populations, to the extent that they show more affinities with middle to early late Pleistocene fossils. What does this mean? It means the researchers compared the 10,000-year-old Nataruk mandibles to a massive global data set, including late Pleistocene North Africans, Holocene Africans, modern Africans, Europeans, Asians, Oceanians, and Arctic populations to determine where Nataruk fits within worldwide human variation. The results are striking. Nataruk shows traits also found in Neanderthals and matches other early Holocene Africans like Assi El Habiod and Iwo Eleru, but not recent sub-Saharans. How interesting. Nataruk aligns closely with late Pleistocene North Africans, not with modern sub-Saharans. The data show that early African populations were far more diverse than today, and that modern sub-Saharan morphology only appears after about 8,000 years ago. Ancient sub-Saharans prior to the Holocene simply do not resemble modern sub-Saharan populations. Let's talk about the 23,000-year-old Lukenya Hill specimen known as KNM-LH1. KNM-LH1 is an East African Kenyan sample dated to roughly 23,000 years. It is the only hominin cranium found with a last glacial maximum late Stone Age assemblage from Eastern Africa. Thus plays an important role in our understanding of the morphology of these populations. KNM-LH. One is distinct from recent Africans and Holocene late Stone Age samples. It is also distinct from most of the comparably aged European Upper Paleolithic samples, except, interestingly, Pestera Q. oase, the oldest EUP cranium. Pestera oase 2 predates KNM-LH1, and since the writing of this paper in 2014, there have been multiple other EUP specimens discovered, but here we have yet another case of a sub-Saharan specimen KNM-LH1 at roughly 23,000 years old, showing no affinity to recent sub-Saharan populations or even to 8,000-year-old Holocene sub-Saharans, but rather affinity with an European Upper Paleolithic sample twice its age, Pestera Q. oase 40 to 41,000 years old, perhaps an example of inter-Africa. KNM-LH1 and other Pleistocene African specimens, all of which are potentially sampling candidate populations for dispersals across and out of Africa during the late Pleistocene, differ substantially, not only from recent Africans, but also from individuals drawn from Holocene late Stone Age archaeological sites. KNM-LH1 and other Pleistocene African specimens found with Middle Stone Age and Late Stone Age artifacts are also distinct from most European Upper Paleolithic individuals. If the Out of Africa model ever had a chance to stand, this would be it, but not so fast. KNM-LH1. As noted by the authors, differs substantially from recent sub-Saharan populations. It does not resemble current sub-Saharan blacks at all. Instead, it shows some similarity with the European Upper Paleolithic Pestera Q. oase, which is dated to 40,000 years or otherwise 20,000 years older than the sub-Saharan KNM-LH1 sample. Furthermore, KNM-LH1 postdates the supposed out-of-Africa dispersal by roughly 50 to 70,000 years or more. This means that whatever populations may have had the opportunity to migrate out of Africa did not resemble present-day sub-Saharans. Instead, the evidence is more consistent with population turnover and inter-Africa movements based on the sample rather than continuity from modern sub-Saharans. Even sub-Saharan Holocene groups differ from the individuals drawn from Holocene late Stone Age archaeological sites. What does this evidence say about the Afrocentric claim of blacks possessing the greatest diversity in the world? The data tells us that much of that diversity derives from ancient populations that bear absolutely no resemblance to modern sub-Saharans. It also reinforces the point that if an out-of-Africa dispersal occurred at all, it was not driven by populations comparable to present-day black sub-Saharans. Early African fossils do not resemble recent Africans. Let's take a look at the PCA and drive home the point exposing Afrocentric bogus narratives. The recent modern human range extends the entire spectrum of European Upper Paleolithic samples, but almost none of the late Pleistocene African samples or the early modern human samples from Africa, Middle Pleistocene, Homo samples from Africa, or the extremely young Iwo Eleru sample, the KNM-LH one sample, which is still young, and almost Hofmeyr sample, which is young enough to cause pause. All these African samples are different when compared to recent modern humans. Why do these older African samples cluster with Neanderthals and not recent African samples? Likewise, why do the recent African samples cluster with European Upper Paleolithic and not the older African samples? Another point of interest is that the NEOAF, or Neolithic fossils from North Africa, are within the recent modern human spectrum, yet cluster in quadrant closest to the dissimilar African and Neanderthal samples. Late Stone Age samples dated from 70 to 20,000 years are more broadly distributed. Recall, late Stone Age samples differ substantially from recent sub-Saharans. Any other study with a PCA scatter plot with this structure would indicate that all recent modern humans are descended from either European Upper Paleolithic or Late Stone Age Africans. But given that the Neolithic fossils from North Africa cluster close to the dissimilar Africans and Neanderthals, that the Iwo Eleru sample is a young 13,000 years old, and that both Hofmeyr and KNM-LH one falls squarely within the European Upper Paleolithic Late Stone Age temporal window. Taken together, these observations support a more conservative interpretation. European Upper Paleolithic and Late Stone Age Africans represent the same population, while the Neolithic fossils from North Africa reflect relatively recent introgression with an extant North African population such as Ibero-Maurusians. The data is further evidence that modern sub-Saharans are the result of admixture between at least two distinct ancestral groups, a Eurasian-derived population comparable to Peshtera, Ku-Oase Upper Paleolithic European humans that migrated into sub-Saharan Africa, and one or more deeply divergent sub-Saharan populations that persisted in the region until relatively recently, contributing 2 to 19% ghost archaic DNA. Within this framework, the high genetic diversity observed among modern sub-Saharans is not the result of simple ancestral continuity from a single source population, but rather the outcome of later admixture between highly divergent lineages. This interpretation is consistent with evidence for archaic introgression within Africa and with documented morphological discontinuities between late Pleistocene, sub-Saharans and recent sub-Saharans, all without invoking ad hoc assumptions about extreme demographic bottlenecks. The Iwo Eleru skeleton is the only Pleistocene human fossil currently known from Western Africa. The Iwo Eleru mandible is most consistent with the shape variation found in North African Homo sapiens from Epipaleolithic contexts, both in overall shape and in its large size. Quote, "Our analysis suggests affinities between the Iwo Eleru mandible and the Epipaleolithic North African sample from Afalu Tafaralt in both its overall shape and size." Iwo Eleru's affinities to the North Africa Epipaleolithic may indicate population movement across the Sahara close to the time of its life, perhaps linked to favourable climatic intervals. These results are similar to previous findings for the early Holocene mandibular samples from Nataruk, Kenya, which were also found to exhibit large size and overall shape affinities to North African Pleistocene and Epipaleolithic samples. Iwo Eleru fell outside the convex hulls of the two sub-Saharan samples, South African Holocene hunter-gatherers and South African recent. Now, why do you suppose a 13,000-year-old sub-Saharan specimen would show stronger affinity to North African Tafaralt than to South African Holocene or recent South African populations? It suggests that Iwo Eleru may represent a population with mixed ancestry, partly related to a North African lineage like Tafaralt, and partly influenced by an archaic ghost population within sub-Saharan Africa. This raises the question of whether this is connected to the deeper ancestry that eventually feeds into lineages such as E1b1a. Another study on Iwo Eleru states that two Iwo Eleru skulls from southwestern Nigeria possess neurocranial morphologies intermediate in shape between archaic hominins and modern humans, as well as the late Stone Age Ishango skulls from the Congo region. Next. What does the science say about the late Paleolithic Jebel Sahaba series? Let's see. Dentally, Jebel Sahaba possess large teeth with high frequencies of mass additive traits and retains archaic cranial characteristics with pronounced robusticity. Jebel Sahaba is an outlier in the later biological diversity spectrum of the Nile Valley, mostly in comparison with Neolithic and later populations. On the contrary, with high frequencies of mass additive non-metric traits, affinities were rather found between the late Pleistocene individuals and historic sub-Saharan populations. This means there was discontinuity between Jebel Sahaba and Neolithic Nubians, meaning there was no affinity with the Nubian A group or ancient Egyptians. Jebel Sahaba was a 13,000-year-old population, still demonstrating archaic features, and its closest affinity is with recent sub-Saharans. Jebel Sahaba is an outlier in the latter biological diversity spectrum of the Nile Valley, mostly in comparison with Neolithic and later populations. Affinities were rather found between the late Pleistocene individuals and historic sub-Saharan populations. The data presented here provide further example of the significant differences in dental aspects displayed between the Jebel Sahaba population and Neolithic samples from the Nile Valley. This means, since Jebel Sahaba shows affinity with recent sub-Saharans, that both Jebel Sahaba and recent sub-Saharans have no affinity with ancient Egyptians, Neolithic Nubians, or ancient North Africans, and certainly not Levantines or Natufians.
Okay.
I believe that Black people are native to every continent on this planet. And that is logical if you believe in out of Africa. So if you're out of Africa, then you should.
This is a black supremacist talking point intended to pursue unwarranted racial superiority. Afrocentrics always fall back on this assertion after they've lost a debate about ancient Egypt. So as a result, they go full throttle bonkers. I've seen it too many times. It's a last-ditch attempt, a Hail Mary, if you will, to make the most impossible, sensational, overblown impression imaginable and hope people are gullible enough to accept it. He certainly never made any effort to substantiate his assertion, did he? No. Also, beliefs are not facts. The idea that any single group created all peoples and all civilizations has zero evidence behind it. Sub-Saharan blacks did not participate in the supposed out-of-Africa expansion that populated the world approximately 60 to 70,000 years ago. Sub-Saharans did not give rise to all people on the planet. They did not magically become Europeans, East Asians, Native Americans, or any other population. They did not change color like a chameleon or magically shed tightly coiled hair in favor of silky, straight hair. Sub-Saharans retain archaic genetic lineages that are completely absent in non-African genomes. This fact alone blows away the asinine idea that sub-Saharans or blacks gave rise to everyone. In fact, sub-Saharans are right where they have been mixing with archaic and moderns in sub-Sahara long after the supposed out-of-Africa event. They never left and are not native to any continent but their own, and their own inner diversity shows this. Carriers of haplogroups A, B and L, including the population proposed to have migrated out of Africa, did not resemble present-day sub-Saharans. This is because modern sub-Saharans carry up to 19% archaic ghost DNA, which was acquired around 40,000 years ago, long after the supposed out-of-Africa event. This means present-day sub-Saharans are not genetic replicas of the ancestral population that left Africa 60 to 70,000 years ago. Sub-Saharan genomes were shaped by introgression from now extinct archaic ghost lineages within Africa, including pulses of Eurasian DNA and isolation, meaning they represent a much more derived and regionally specific genetic profile. As witnessed in this video and studies presented herein, present-day sub-Saharans look nothing like their pre-Holocene predecessors. Early sub-Saharan samples from before 8000 BC display traits associated either with into sub-Sahara gene flow or with broader, globally distributed human variation, depending on the specific sample. In addition, Eurasian populations do not carry the ghost population ancestry component found only among sub-Saharan groups, which ranges from roughly 2 to 19%. Haplogroups A, B and E, 1B, 1A are geographically restricted to sub-Saharan Africa and are absent in Eurasia. Nor do Eurasians carry the MUC7 allele or H-PAL restriction site polymorphism at nucleotide position 3592. The oldest Eurasian skeletal morphologies do not resemble any African skeletal morphologies at the supposed timing of the out of Africa fantasy or Rugli 70 to 100,000 years ago. Almost all black African populations possess the two haplotypes GM1, 17, 5, 6, 10, 11, 14, and GM1, 17, 5, 6, 11, 24, generally not found in other populations which appear to lack allotype G3M6. This also characterizes black Africans. Sub-Saharans possess the H-PAL site gain at nucleotide position 3592 within the mitochondrial DNA genome. This marker is completely absent among non-African populations. Assuming that phonetic expression approximates genetic variation, previous dental morphological analyses of sub-Saharan Africans by the author show they are unique among the world's modern populations. Numerically derived affinities, using the multivariate mean measure of divergence statistic, revealed significant differences between the sub-Saharan folk and samples from North Africa, Europe, Southeast Asia, Northeast Asia, and the New World, Australia, Tasmania, and Melanesia. Did you hear that? Significant differences exist between sub-Saharans and the rest of the world's populations. You see, non-Africans did not descend from sub-Saharans, and there's no evidence that they did. What else does it say? Sub-Saharan Africans are characterized by a collection of unique mass additive crown and root traits relative to these other world groups. The most ubiquitous of these traits are also present in dentitions of earlier hominids, as well as extinct and extant non-human primates. There it is, extinct and extant non-human primates. We are not alike. What else does it tell us? Sub-Saharan Africans differ markedly from all others. Do you hear this? Sub-Saharans differ markedly from all others in their recurrent expression of such complex mass additive traits such as Bushman canine, two-rooted UP1, the UM1, Carabelli's trait, and so on. But what of the UM1, Carabelli's trait? The UM1 Carabelli's trait is present in Australopithecus afarensis, Australopithecus africanus, Australopithecus robustus, Homo habilis, and Homo erectus. It is also present in living gibbons, chimpanzees, and gorillas. Africans are genetically clearly differentiated from other populations of the world. They do not seem to constitute the latter's direct ancestors. There it is. Africans, or in other words, sub-Saharans, do not constitute as direct ancestors of other populations. That is why we favour the hypothesis of non-African populations colonising Africa after having been subjected to a drastic founder effect and random genetic drift. Matthias Jacobsen et al. 2025 informs us that many of the ancient southern Africans, including those who lived between about 10,200 and 1,400 years ago, fall outside the range of genetic variation among modern-day individuals and form an extreme end of human genetic variation. The researchers labelled this previously unknown suite of genetic variation the ancient Southern African ancestry component and found that there was no clear indication of admixture or outsiders sharing their genes with the group until about AD 550. Quote, all ancient Southern Africans dated to more than 1,400 calibrated before present show a genetic makeup that is outside the range of genetic variation in modern-day humans, including Southern African Koisan people, although some retain up to 80% ancient Southern African ancestry, manifesting in a large fraction of Homo sapiens specific variants that are unique to ancient Southern Africans, end quote. So you have sub-Saharan specimens that fall outside of human genetic variation up until the last 1,500 years. Not only that, the oldest of these, 100,000 years, show no indication of genes outside of Africa. So much for the fantasy that is out of Africa. There are significant differences between pre-Iron Age Eastern Africans and contemporary North, Sub-Saharan and Southern Africans, suggesting diachronic changes in African dental complexes. Recent Africans may therefore be poor proxies for the genetic relationships in antiquity. Given this documented temporal and regional variation, claims that any present-day population represents a universal ancestral hierarchy, point of origin, or biological benchmark for ancient civilizations are not supported by the evidence. Are you seeing the bigger picture? Never was, because we never stepped foot out of Sub-Sahara. A study authored by a genetics team in Barcelona, Spain, informs us that sub-Saharan populations formed a well-differentiated cluster from the remaining world populations, grouped in two close clusters, that of Asian populations on the one hand, and that of North African, Southern European, and Middle Eastern populations on the other hand. This is because sub-Saharans carry certain ancestries, including archaic ghost DNA, that non-Africans do not carry. Again, the out of Africa fantasy thoroughly dismissed. Some ideologically driven narratives claim that the Koisan represent the oldest human population and therefore the ancestral source of all humanity. This claim is not supported by genetics or anthropology. While Koisan populations harbour deep genetic lineages, genomic studies consistently show that they are genetic outliers within sub-Sahara, shaped by long-term isolation and population structure. They did not give rise to other populations within sub-Sahara, therefore there is no plausible mechanism by which they could have given rise to populations outside sub-Sahara or Africa as a whole. Neither Koisan Y chromosome lineages nor mitochondrial lineages are found beyond Africa, nor do their distinctive cranial and postcranial morphologies appear in non-African populations. Modern human diversity reflects a complex, structured past. Genetics and physical anthropology therefore directly contradict the notion that the Koisan were the sole progenitors of humanity or the source population for Eurasian peoples. The notion that Europeans, or Eurasians more broadly, are merely a hybrid mixture of sub-Saharans and Neanderthals is likewise unsupported and has been decisively dismissed by anthropological evidence. PCA alone dismisses out of Africa as pure fantasy. The documented presence of Eurasian-associated genetic lineages in North Africa since the Paleolithic, the well-established and persistent Eurasian admixture in Eastern Africa, and the downstream diffusion of Eurasian genetic markers across the Sahara, the Sahel, and into parts of sub-Saharan Africa, including among Bantu and Koisan-speaking populations, stand in sharp contrast to the complete absence of evidence for an ancient sub-Saharan African presence outside Africa. Taken together, these facts seriously undermine the validity of a rigid and oversimplified out-of-Africa narrative. And this leads us directly to Into Africa. Quote, "We find that the method infers high rates of migration from descendants of non-Africans to Africans, but not in the opposite direction, in the period 30 to 70,000 years ago, corresponding to the late Middle Paleolithic." End quote. This is because the population traditionally assumed to have left Africa may have been genetically similar enough that outward migration was not readily distinguishable from return migration using this method. In other words, the source and recipient populations were similar at that time. These populations were differentiated from late Stone Age sub-Saharans and therefore did not resemble either ancient or recent sub-Saharan populations. Let's continue. Quote, "In populations from the Niger-Kordofanian and Nilo-Saharan language groups, comprising the majority of the population on the African continent, the peak inferred migration rate from Eurasian populations most frequently falls in the epochs, spanning 35 to 45,000 years." End quote. This time frame corresponds to when archaic ghost DNA appears to have entered certain sub-Saharan populations. Could it be that this admixture event reflects interactions between populations with Eurasian-related ancestry already present in Africa and local archaic hominins, contributing indirectly to the genetic landscapes from which later Niger-Congo, including Bantu-speaking and Nilotic populations, emerged? Continuing quote, populations in the Afroasiatic language group show evidence of large amounts of directional migration in the Holocene, which is consistent with previous findings of relatively recent European introgression into these populations. In all cases, the migration rates from Eurasia to Africa are substantially higher than in the opposite direction, consistent with the findings in the Simon's Genome Diversity Panel. The contribution of this admixture event to extant African genetic variation is substantial. The estimated integrated migration fraction indicate that for individuals in the major African language groups, approximately a third of ancestral lineages trace their ancestry through the proto-Eurasian population Niger-Kordofanian group and Nilo-Saharan groups. We infer substantial Eurasian backmigration into all African groups. However, the inferred IMFS for individuals from Koisan populations are significantly lower than for any other group, end quote. That's right. The paper explicitly confirms that all sub-Saharan populations have received Eurasian genetic inflow, some to a greater extent than others, but none are exempt from this admixture. We use sequential Monte Carlo inference of the sequentially Markovian coalescent to investigate ancient migration rates and population substructure, and found evidence for a substantial admixture from ancestors of present-day Eurasian populations into African populations in the late Middle Paleolithic. Our analysis suggests that a population ancestral to present-day Eurasians contributed as much as a third of the genetic material in many modern African populations. In light of these recent studies, the observations in this paper add to a growing body of evidence for complex population structure and migration surrounding the out of Africa event, leading to a substantial replacement of the African population in the late Middle Paleolithic.